So I will first give you a general background on malaria, how we came to know what malaria is and some of the prominent features of its epidemiology. And then I'm going to focus on the pattern and pathogenesis of severe malaria. There are many aspects of severe malaria, specifically in African children, that are unusual and certainly have an explanation at a molecular level, although we don't know many of the explanations. And then I'm going to focus on what we've learned from pregnancy malaria. This is an area that my laboratory is focused on and has illuminated some host parasite interactions that explain the epidemiology and also reveal that much of the burden of malaria may be masked. So what is malaria? Well, first of all, malaria is bad air because it was widely believed that bad air is what gave you malaria. People that live near marshes tended to get malaria. And it's been described from the very earliest texts in human written history, dating back to 2700 BC with Nei Ching, which was the Chinese medical text. And it was recognized because of its distinct signature, which are periodic fevers. These are fevers that recur on a regular basis, either every 48 or 72 hours, and also by splenomegaly. So the combination of these is how we recognize these in the ancient literature. However, it became something else, of course, when Alphonse Laveran recognized that there was a parasite infecting a French soldier in Algeria. That was in 1880. And then subsequently Ronald Ross determined that the parasite was transmitted by a mosquito vector. And both those gentlemen won Nobel Prizes for those discoveries. Now here is the malaria life cycle. And it's not to get you into the gory detail. You can see it's a complex life cycle. I will just highlight that there are three major aspects of it. First of all, there is a blood stage to the malaria life cycle here. And as I mentioned, Alphonse Laveran, I recognized this stage of the parasite. But actually there's two forms in the blood. There's an asexual form of the parasite that is undergoing repeated divisions. And it is those repeated divisions and ruptures of the red cell by the asexual form that causes those periodic fevers. Some parasites grow for 48 hours and rupture the red cells, so their periodic fever is every 48 hours. And some parasites take 72 hours to complete that developmental cycle, and therefore their periodic fever is every 72 hours. The form that Alphonse Laveran recognized was actually the sexual form. Some subset of the progeny of the parasites will divert into a sexual pathway. That is the form that gets into the mosquito. And Alphonse Laveran recognized it because it is motile. It's like a sperm, basically. If you look at it, it looks very similar. Then there is, after the blood stage, the mosquito stage, where the parasite actually has to invade different membranes to get into the mosquito and then get back out to get into the human host. And it also takes on an encysted form in the mosquito stage. And then finally there is the liver stage. When the parasite first infects you or me, it actually lives in the liver for a week before it gets into the bloodstream. So that stage of parasite development is silent. People don't feel sick. It was hard to discover. You can see it was 50 years after Ronald Ross identified the mosquito stage that it was finally recognized that the parasite was going to the liver. They actually knew it was going somewhere. They didn't know where, so it took 50 years to find out where that parasite was going. And it was going to the liver. As you can imagine, finding a few parasites in liver is not easy. Those stages, those major stages of the parasite life cycle also explain the different strategies that people are using to try to attack the malaria problem. So in the first instance, people are trying to develop vaccines. You would immunize people, elicit antibodies, and they would be taken up by the mosquito with an infected blood meal. And they would be targeting parasites that the proteins that the parasite would express as it went through its sexual and mosquito stages. So these are proteins actually that the hosts aren't normally exposed to. You would elicit antibodies against them, and then it would arrest the development of the parasite in the mosquito. Those vaccines obviously are not going to help the person receiving the vaccine, but they're oriented to the community, and they will block transmission. And there is evidence that these vaccines are feasible in animal models. Then there are vaccines that would target this liver stage of the malaria parasite. And the intention here is to completely block infection. And we know it's possible because we have a model that tells us it's possible. If you attenuate the parasites that are in the mosquito before it delivers it to the human host, and attenuate those in a way that they arrest during liver stage development, those parasites will elicit a highly effective immune response that have been shown to protect mice and humans from subsequent challenge with wild-type parasites. But it takes a lot of them to induce that immunity. And so people are trying to develop subunit vaccines targeting specific proteins expressed by those liver stage parasites that might induce a similar kind of immunity. The intention there is to completely block infection. If you don't completely block infection with that vaccine, at least in simple terms, it doesn't seem like it would do you much good because it gets through to the blood stage, and blood stage can multiply to a level where it will cause disease and death. And so then there are vaccines that are intended to block disease and death that target the blood stage, and I'll tell you more about that in the course of my talk. So where is malaria today? This is from a paper that just came out a couple of months ago. It's in February 2008. And this has been a long-standing problem with malaria because the places where people really suffer from malaria are the places where we have our poorest infrastructure for collecting vital information about people's health and their deaths. What they've done this group has done is to take some well-characterized sites where we do have a lot of information about the incidence of parasitemia and disease due to malaria, and then extrapolated that to other areas where the climatic conditions are similar in a way that you would expect that the incidence of malaria would be similar in those communities. And so it's, there are 87 countries that are affected by malaria. There are 2.4 billion people that are at risk. And the estimates are that there were 500 million cases of clinical malaria last year. That's many more infections than that because many people that are infected don't know they're infected. But 500 million clinical cases and 1 million deaths, of which most of those are going to be under 5 children in Africa. And where was malaria yesterday? Well, malaria used to go widely outside tropical and subtropical zones. As you can imagine, malaria flourishes in subtropical and tropical zones because mosquitoes do well there, but it's also that the parasite develops well in the mosquitoes there because the parasite's development itself is sensitive to the climatic conditions, including the temperature. But as you can see here, dating, if you go back to 1900, and that's what these very light pink zones indicate is where malaria was in 1900, it extended far into temperate areas. A lot of this was a specific parasite form, Plasmodium vivax, because Plasmodium vivax can develop at a lower temperature in the mosquito than Plasmodium falciparum. But there was also Plasmodium falciparum in some places that you wouldn't expect it. And you can look here in our own country that up until 1946, it was widespread in the southeastern United States. And it was finally eradicated there in 1951. The incidence of it had gone down steadily, first with the Tennessee Valley Authority, which organized the water systems in the southeast. And then during World War II, there were real efforts to control malaria around military installations in the southeast. But finally, there was a 4-year program which was led by the CDC using DDT and indoor residual spraying that finally eradicated malaria in the southeastern U.S. Now, those places where malaria used to be are very different from where the places where malaria is now. Where malaria used to be, it was very seasonal. It would tend to have a marginal transmission. And when it gets to a certain level, that level of transmission is called unstable because it's so sporadic that very few people are affected by it, but it can turn into an epidemic. And so you'll read historically, when you read historically of epidemics, those are occurring in places where it's typically an unstable transmission area. In much of the places where malaria remains, it is fairly stable transmission. That means typically there's malaria transmission every year and many people are being exposed to it. As a consequence of this, the epidemiology is very different in these two areas. And even today we have areas of unstable transmission, low, and stable transmission, which is high transmission. The main reason that these areas are different is because people develop immunity that tends to control malaria. And for that reason, the distribution of malaria, but also the pattern, the syndromes that people experience are very different. So in an area of unstable, low transmission, there's not very much immunity that's acquired. So what you see is that people that are getting malaria and presenting that are sick are spread across all ages. When they do get malaria, it is at high risk of becoming severe malaria. And typically the syndromes that these individuals experience and what we would experience when we get malaria is multi-organ disease. It typically involves many organs. In areas of stable transmission, there's more infections, so immunity is acquired and it's maintained by continuous exposure. Therefore, most of the disease that you visibly see is in young children. If a child in the communities where we work lives to be 3 or 4, they're very unlikely to die from malaria. It's also true that the syndromes they present with are very discrete syndromes. So whereas a person in a low transmission area, an adult person would have multi-organ disease, they have discrete conditions: cerebral malaria, respiratory distress, and severe anemia. Now this is the epidemiology of Plasmodium falciparum malaria in a typical African community. As I'll explain to you later, Plasmodium falciparum parasites are the most virulent. They're the ones that cause the vast majority of deaths due to malaria. And I want to highlight a few things. First of all, if you know anything about malaria, you may know that it takes a long time to become immune to malaria. So what this red line is indicating is over the early years of life, the incidence of different manifestations of malaria, and in this case the red line indicates just how frequently you have parasites in your bloodstream. And so after a period of 2 or 3 months in these communities where neonates have something that makes them resistant, we're not exactly sure what it is, but certainly maternal immunity is contributing. Infants become susceptible to malaria, parasitemia, and they will remain susceptible for several years. Typically between 5 to 10 years, you'll start to see the frequency and the density of parasitemia go down in those children. And then particularly across adolescence, you will see it go down to an adult level of control, where really adults present infrequently with parasitemia. When they do, it's a low-density parasitemia. That, of course, has made people pessimistic that a vaccine is possible because of the idea that the parasite's evading the immune system. However, what's striking is that the pattern of resistance that people acquire to severe malaria is very different. In fact, there's evidence that kids may only have 1 or 2 episodes of severe malaria before they acquire some kind of resistance that prevents that from recurring. And that's why children stop getting very sick and dying long before they start getting parasitemia. But it also tells us that whatever it is that controls severe malaria is something different than what controls parasitemia. And then finally, I want to highlight a distinct aspect of malaria, which is malaria during pregnancy. So, of course, by the time a young woman attains the age of maturity, she's equal to her counterparts at that age in terms of her immunity, her ability to control parasitemia. But when she has her first pregnancy, she becomes susceptible again. And she is more likely to be infected. If she has marginal immunity, she's more likely to get severe malaria. And if she gets severe malaria, she's more likely to die than her non-pregnant counterparts. However, over a couple of pregnancies in the communities where we work, you will see that women become resistant again to malaria. Now, where are all the deaths occurring? Well, as you might expect, a lot of deaths are occurring in African children under the age of 5 because they're having these episodes of severe malaria. But another place where deaths are occurring is due to pregnancy malaria. There's quite a number of deaths of mothers from malaria, and I'll give you some features that explain that it's really under-recognized how much damage malaria is doing to mothers. But also, it's estimated that the effect of malaria to cause low birth weight or premature deliveries is contributing to infant mortality. It's estimated that 100,000 African infants die each year from low birth weight. Another interesting and important aspect of malaria mortality is that any place people have eradicated malaria, the effect on overall childhood mortality has been greater than one would have predicted based on what people thought malaria was doing to kids. So there is direct malaria mortality. I told you kids present with cerebral malaria, respiratory distress, severe anemia. You'll see parasites in their bloodstream, and the inference is that this is malaria causing death. When people have made estimates of how much malaria is contributing to childhood mortality in African communities, they typically come up with figures of about 20 to 25% of childhood mortality in those communities is due to malaria. But then there's something called indirect malaria mortality, and it's something that we don't understand. The idea is that somehow malaria contributes to deaths. Maybe it's assigned to another organism, but malaria is present at the same time. I think our evidence for that is not strong. There are some comorbid conditions, some comorbid infections that probably contribute to severity of malaria, but I don't think it explains that. However, when efforts have been made in communities to eliminate malaria, primarily using residual insecticides, to the point where you really nearly eradicated transmission in that community, what they've seen is that it reduced childhood mortality 40 to 50%, which would leave you to infer that this indirect malaria mortality may be 20 to 25% of childhood deaths in those communities. So let me talk a little bit about the pattern and pathogenesis of severe malaria. So this is another illustration of the parasite's life cycle. Here is the mosquito stage. Here is the liver stage. And here is the blood stage when people get sick and die. There are commonly known to be 4 human malaria parasites. Plasmodium falciparum, I've told you, is the deadliest. Plasmodium vivax is the one that was often common in temperate climates. Plasmodium vivax and Plasmodium ovale have the property of being able to take a dormant form in the liver, and that's why they can recur months or years later as a relapse coming from the liver. Plasmodium malariae is the 4th human malaria parasite. It has the property not of being dormant in the liver, but actually developing a form that can persist for decades in the bloodstream. They've had cases where somebody left a malarious area, had surgery, 50 years after leaving, last visiting a malarious area, and Plasmodium malariae recrudesced in those individuals. So when malaria comes back from a blood stage infection, it's called a recrudescence. When it comes from a liver stage infection, it's called a relapse. More recently, it's been recognized that this parasite, Plasmodium knowlesi, that was first recognized as an infection of macaque monkeys, is actually naturally transmitted to humans in some areas, such as Malaysia. So there's increasing evidence that Plasmodium knowlesi may be our 5th human malaria parasite, but it specifically has a reservoir in macaques. There's not knowledge that the other 4 human malaria parasites have a non-human reservoir. Now, why does Plasmodium falciparum, why is it specifically virulent? It's believed that it's so deadly because of its property to express proteins on the surface of the infected red cell that allow the infected red cell to bind to receptors on the endothelium and sequester in large numbers in deep vascular organs. This is an example of that, where what you see in this cross-section of a cerebral vessel are 3 infected erythrocytes, as well as an uninfected erythrocyte. And so these are 3 parasitized red cells that are sequestering in a cerebral vessel, and this person died of cerebral malaria. And this is an image of an uninfected red cell, which you see there. This is an EM image of an infected red cell, and what you should appreciate is the stippled pattern all around the surface of the infected red cell, and those are actually small protrusions that are electron dense that you can appreciate here as being electron dense on the surface of the infected red cell, and those actually become points of contact between the infected red cell and the endothelial surface. There are proteins or molecules that are mediating the attachment of the infected red cell to the endothelium. So of course it's been of great interest to identify what those proteins are, because maybe by interfering with those we can prevent virulence with Plasmodium falciparum. And what we've learned over the past 10 years is that there is this complex variant antigen family called PFE and P1 that's encoded by the multi-copy VAR genes. There's about 60 VAR genes in every parasite's genome. And this just shows the structural representations of all the VAR genes, the encoded PFEMP1 proteins from our prototype parasite 3D7. What you see in color are the extracellular domains of that protein. So what you can appreciate is that these proteins have several domains. They can vary extensively in their domain architecture. You can tell just by the coloring and the patterns here. Many of them look similar, but many of them look very different. I will also tell you these are very large proteins, and that you can see diversity within a single genome. There's also extensive diversity between genomes, so that to recognize one of these from one parasite to another is not common. I will give you an example of one that is common, and it has a very special function, we think. And this is just a closer look at two of these, but it points out that in the studies that people have done since these were discovered in 1995, there's been extensive evidence that recombinant forms of these domains that constitute PFEMP1 can bind to many of the endothelial receptors that have been recognized to support and mediate parasite adhesion. So there's evidence, there's good evidence that PFEMP1 proteins are exported to the surface of these knobs, and there's in vitro evidence that these recombinant forms of these can bind to the proteins on the endothelial surface that are supporting parasite binding. And therefore these have been a very attractive target for intervention, except for the fact that they're so highly variant. So what you can imagine in this scenario, each infected red cell only expresses a single PFEMP1. So I've told you that the different domains bind to different molecules, different endothelial receptors, and different PFEMP1s have different domains. Therefore, you can imagine that depending on what PFEMP1 molecule you put on your surface, you would bind to a different receptor. And certainly we see that we can get parasitized red cells that bind to different receptors. However, that leads to a model where perhaps what's happening is by varying the PFEMP1 that's on the surface, the parasite can vary where exactly it's going to bind to. And if the endothelial receptors differ between vascular beds, that means that depending on what the PFEMP1 molecule that the parasite's expressing is, it will go to one or a different bed. Now, PFEMP1 is also highly immunogenic. It elicits antibodies against that PFEMP1. By developing those antibodies, the parasite would have the effect of causing the parasites to select against that particular PFEMP1. So you see two parallel functions potentially for PFEMP1. One, it is an immunodominant protein, and that as the host develops immunity against that PFEMP1, the parasite will put a different PFEMP1 on its surface. So it would stay ahead of the immune response by doing that. But in parallel, the PFEMP1 is allowing the parasite to bind to endothelial receptors and sequester in vascular beds. We believe that's important because Plasmodium falciparum always does that. It may be because it prevents the parasite from being cleared in the spleen, but somehow the parasite has had to resolve this need to vary this major surface protein and at the same time to exploit it to bind to receptors that it needs to sequester in endothelial in vascular beds. And we are still working out how the parasite might be able to do that. So a simple model of malaria virulence is that severe malaria is mediated, or related at least, to parasite adhesion and sequestration, and that PFEMP1 is the major variant surface antigen on the surface of the infected red cell, and that it mediates parasite binding and therefore virulence, but that you can develop immune responses against the different PFEMP1s that would cause the parasite to select a different variant to put on its surface. So the first thing that probably comes into your mind is if we could just figure out the PFEMP1 that's causing, for example, the parasite to sequester in the brain, we'd have a vaccine, and certainly that is the thought that we have. We've been unable to identify that, and in fact we don't yet know what the receptor in the brain is for the parasitized red cell. Now, I told you that in African children, severe malaria presents as discrete syndromes, and here's some data that supports that. So what you see is this is work by Kevin Marsh and his group in Khalifi. This blue, this is a Venn diagram. The blue are children with cerebral malaria. The red are children with respiratory distress. The yellow are children with severe anemia. And what you can appreciate is that actually there's more cases that have the discrete syndrome than an overlap of these syndromes. And it's actually really not fully fair because a lot of kids that develop severe anemia is hemoglobin less than 5. Of course, they're going to develop respiratory distress. So many of these cases of respiratory distress are actually due to severe anemia, but there's a whole separate syndrome of kids that have respiratory distress and have normal hemoglobin. It's associated with metabolic acidosis. And all of these are deadly syndromes, but the mortality is highest in the kids that do have an overlap of these conditions. They are in a worse condition. But the common presentation is as a discrete syndrome. It's also true that the epidemiology of these severe syndromes in children differ. And one way that they differ is that they peak at different ages. So what you can see here is the peak incidence of severe anemia in a low transmission area like Senegal or a high transmission area like Kenya compared to the peak incidence of cerebral malaria in those places. So one thing that you can appreciate, first of all, the peak incidence is younger in a high transmission area. That makes sense. I told you that in high transmission areas, people develop immunity actually relatively quickly. So it makes sense that actually the peak incidence is younger in Kenya than it is in a low transmission area like Senegal. But what you can also appreciate is that cerebral malaria is peaking, its peak incidence is occurring at an older age than severe anemia is both in Senegal and also in Kenya. I just showed two sets of data here, but it is a consistent finding between areas. The other thing is that the proportion of severe malaria cases due to cerebral malaria varies with the intensity of malaria transmission. So Bob Snow and Kevin Marsh took data from many communities across Africa. On the left, you see the lowest transmission area. On the right, you see the highest transmission area. And then you see just increasing intensity as you go along in these different communities. And what you can appreciate is that cerebral malaria as a proportion of severe malaria cases goes from almost nothing in an area of very high malaria transmission to 50% or more of the severe malaria presentations in a low transmission area. So what could be causing kids to have different, very distinct syndromes in a community? Some kids get malaria, they come in with severe anemia, some come in with respiratory distress. Some come in with cerebral malaria. And then also, what could explain differences in age? I'll tell you, we really don't know. But of course, one thing that importantly modifies the risk of severe malaria are genetic factors. I'm sure you're familiar with sickle trait, but there's quite a number of red cell abnormalities that confer protection from severe malaria and death due to malaria. These are listed here. There's also a number of immune polymorphisms, immune gene polymorphisms that have been associated with resistance to malaria. And in fact, malaria has been the strongest selective force on the human genome over recent history. And how might these red cell abnormalities influence your likelihood of getting severe malaria and dying? Here's one example of how it might work. I showed you these images of an uninfected red cell here and an infected red cell a few slides ago. You can see the infected red cell has this stippled knobby pattern that the parasite uses to bind. What you can see is that when parasites infect a red cell from a child that has hemoglobin C, that actually the knob distribution is very different and the knob appearance is very different on the infected red cell from a child with this genetic polymorphism. These parasites also don't bind very well. They don't adhere to common receptors very well. And so the suggestion is that maybe these genetic polymorphisms in red cells may be impaired in their ability to adhere to endothelium, and that reduces your likelihood of getting severe disease and dying. And this group, Fairhurst and Wellams, has recently provided evidence that hemoglobin S, similarly, will modify the parasitized red cell's ability to adhere to endothelial receptors. One interesting feature that we've seen, so you see that there's a whole panel of different resistance genes, it turns out that these don't work in isolation. They actually interact with each other. And the ways that they interact are not always favorable to the host. So what I'm showing here is some data that was collected in Kenya. It's a little complicated, but basically it is showing the incidence of 3 different things: uncomplicated malaria, malaria that caused the child to go into the hospital, and severe malaria. And they've normalized all the data to normal children. So here you see these are children that do not have thalassemia and they do not have sickle trait. That's this group right here. And so in this first group, you're seeing children that have different degrees of alpha thalassemia on a background of AA hemoglobin. And here you see, again, children that don't have thalassemia or have single or double deletions on a background of sickle trait. And so what you can see is that actually thalassemia is pretty protective from severe malaria. This is a double deletion on a background of AA, and the likelihood of presenting with severe malaria, the incidence of it is about 50% of a normal child. You'll also appreciate how effective sickle trait is. It has almost no risk of getting severe malaria among the children that were in this cohort compared to the children with the normal genotypes. Of course, that huge advantage is balanced by the fact that any child that gets 2 copies of this, there are no adults in these communities that have sickle cell disease. All individuals that get sickle cell disease die at an early age in these communities. But what you can finally see is that children that have both of these abnormalities, and that's indicated here, have both the double deletion alpha thalassemia as well as sickle trait, confers no benefit. They are at the same risk of getting severe malaria as the children who have a normal background. So there's an interaction between these between these 2 polymorphisms and likely between other ones that might explain some of the complexity that we see in trying to interpret why children present with different syndromes or a different risk of severe malaria. And we've seen that ourselves in Tanzania. Where we work in Tanzania, the malaria transmission is very intense. There's something called an entomologic inoculation rate, which is an estimation of how frequently you're bitten by an infected mosquito, how frequently parasites are being inoculated into you. And in the community where we work, the EIR is 400, which means that on average you're getting 1 infected mosquito bite every night. And despite the fact that there's such a saturation of malaria exposure in this community, children can have very, very different experiences with malaria. So what we do in our studies is we recruit children at the time of birth. We take blood smears every 2 weeks from them to quantify how frequently they're getting parasitemic, and then we also monitor their illnesses. What you can see in these figures, this is one child over the first 2 years of life, and this is another child over the first 2 years of life. The blue squares are actually the blood smear results, whether there were any parasites in the blood smear every 2 weeks that we looked at the child. And the red is the child's hemoglobin, where the first red line is the definition of moderate anemia. And what you can appreciate in this child that we followed for 2 years, this is increasing age, is that just about every 6 weeks this child had parasites in his blood smear. Whereas this child never had a parasitemia during the entire 2 years that we saw him. And in fact, about 8% of the kids in this community with intense malaria exposure never presented with a parasitemia, despite the fact we were actively following them. These children were born 1 day apart. They don't have any of the known malaria resistance genes. Whether there might be other resistance genes we're not aware of, we don't know. But it just highlights the fact how different the experience of these children, the experience these children can have in areas of malaria transmission. Now I want to highlight some of what we've learned from pregnancy malaria, which has been the area of interest for our laboratory. And the first thing I'll talk about is really what we've learned that explains the epidemiology as well as the basis for immunity. So just as I told you that the pattern of malaria is different in areas of stable and unstable malaria transmission in other individuals, it's also true in pregnant women. In low transmission areas where women won't have had much immunity, when they get malaria during pregnancy, It is typically severe. It's more severe than in their non-pregnant counterparts. It's likely to kill them, and it also has deleterious effects on the development of their fetus, leading to low birth weight with consequent infant mortality. And it's also appreciated that there's a lot of abortions and preterm deliveries in these women. In a high transmission area, women have high levels of systemic immunity. They actually often will not be aware that they're infected, and therefore it becomes an insidious disease. It often leads to severe maternal anemia, which develops as a chronic process in women who are infected, and this also leads to low birth weight and neonatal infant mortality. So despite the fact that women aren't presenting symptomatically typically, they're actually increasing their risk of mortality both for themselves and their offspring. And I told you that malaria susceptibility is greatest in first pregnancy. This is just data that we collected in a high transmission area of Kenya, and this just shows the prevalence of malaria parasitemia in first-time mothers, second-time mothers, and third-time mothers in this community, showing that women are acquiring resistance. If I showed you the parasite density in these placentas, it would really highlight the fact that it is these first-time mothers who get quite intense placental malaria. And this is placenta malaria. This is an image from a fresh frozen section that we prepared. And what you can appreciate, the tissue here in the placenta is fetal tissue. These are fetal cells in the placenta. And then in the mother's vascular spaces, what you'll see is the accumulation of these parasitized red cells. The dark brown that you see there is the parasite pigment that the parasite accumulates by digesting hemoglobin. And you can see that the density of these parasites is actually quite high in the maternal vascular spaces. So what we learned, we being Mikhail Fried and myself, when we went to work in Kenya, we had the hypothesis that what would explain the women's epidemiology and that first-time mothers were susceptible and third-time mothers were resistant would be that this could be explained by a distinct parasite phenotype that women were acquiring immunity to over successive pregnancies, and that's basically what we found. So the first thing that we learned was that the parasites that infect pregnant women bind to something different than the parasites that infect other people. This was the first evidence we have. This is an assay that we developed where we took sections from uninfected placentas, collected parasites from infected placentas, and looked to see if they would bind on to the uninfected placentas. And what you can appreciate here is that in this assay, parasitized red cells bind right along the surface of the placental lining. Again, these are fetal cells in the placenta, and the mother's infected red blood cells are binding to the surface of that. If you take parasites from non-pregnant individuals, they don't have that property. And we've learned in the course of those studies that what the parasite was binding to was chondroitin sulfate A. This is a glycosaminoglycan that the fetal cells express. It's both on the surface of the fetal cells, and it's also distributed in the maternal vascular spaces. And that would explain why the parasitized red cells actually distribute throughout the maternal vascular spaces. This binding pattern is very different from the binding pattern that we see in parasites from non-pregnant individuals, which bind to CD36 routinely, but do not bind to CSA. We subsequently learned that women developed antibodies against these parasites in a way that would explain their epidemiology. So in this assay, we take sera from pregnant women, first-time mothers, second and third-time mothers, and look to see the degree to which they inhibit binding. And what you can appreciate is that the level of these antibodies in first-time mothers is very low. The level of these antibodies in multigravid women is very high, consistent with the epidemiology. And of course, this has become the basis for developing a vaccine, because we'd like to identify what those antibodies are recognizing on the surface of the infected red cell. And this is a project that's supported by the Gates Foundation. The parasite has over 5,000 genes in its genome. And over the past 5 years, we've worked with partners from Copenhagen, France, and in Tanzania to use a number of different strategies to identify what is different about these placental parasites. And we've identified a single PFEMP1 form that is typical of the parasites that infect pregnant women. So 1 out of those 60 genes that I told you about. It also turns out that this is one that you can see in every parasite that you look at. So it's unusual in that it is a variant antigen gene, but it is conserved in different parasites. We've also found that there are other proteins that are not from a variant antigen family that also are expressed by the parasites that infect pregnant women, but not the parasites that infect other people. And so at this point, what we are doing now is to try to develop these as immunogens, expressing these as recombinant proteins, immunizing mice to see if we can develop these anti-adhesion antibodies that women naturally develop. And so to explain malaria in pregnant women, what we found was that the parasites of pregnant women are distinct because they bind to a different receptor in the placenta, and that women become resistant to pregnancy malaria as they develop antibodies against placental parasites. We found that there's a single VAR gene form that's expressed by these placental parasites, and this is our lead candidate for a vaccine, but we're also looking at some of these other proteins that pregnancy parasites express. And the concept is that a vaccine would be given to women before their first pregnancy, and then they would develop an immune response that would be naturally boosted because that's what happens in their second and third pregnancies anyhow. So finally, I want to talk a little bit about the masking of disease burden, how we've learned from pregnancy malaria, and it makes us think that maybe we can take some of these lessons and apply them to why it is that so many more children are dying from malaria than we can appreciate when we just try to follow the parasite. We've been doing studies in Tanzania, as I mentioned, where we follow kids from birth. We also intensively characterize the pregnant women who agree to have themselves and their child participate. And these are the two communities where we're working. This is where Muheza is, and this is Morogoro, two communities that we work with in Tanzania. Our interest really here has been the interface between pregnant women and the long-term outcomes for their children. We had an appreciation for the fact that pregnant women were infected by this different parasite. We've also been interested to try to get an idea of what are all the different factors that are affecting malaria susceptibility in pregnant women as well as in children. We looked at a number of these in the pregnant women who presented and agreed to participate in our study. We looked at whether the mother was presenting in the low transmission season or the high transmission season, whether she used a bed net, whether she took drugs during pregnancy, whether it was her first or later pregnancy, and then also whether she had a normal iron status or was iron deficient. And when we did all of these in a multivariate analysis, actually these factors that you would have thought would have an impact on her risk of having placental malaria delivery did not have a significant effect. The things that had a significant effect were actually whether she was a first-time mother or a later mother, and then also whether she had a normal iron status or was iron deficient, where iron deficiency actually conferred resistance to placental malaria. And this is what those data look at when you stratify by the iron status and the mother's gravidity. What you can see is that actually iron deficient women in first, second, or third time mothers, their likelihood of having placental malaria at delivery is significantly lower than the likelihood of women who have a normal iron status. Which is bad news because all the women that we see are getting iron whenever they come in for an antenatal visit. And I'll tell you, I won't show the data, but there's just been a study completed 2 years ago in Tanzania on the island of Zanzibar, where children who got iron supplementation actually had significantly increased mortality related to malaria. So I actually don't know what we're doing for women by giving them iron during their pregnancy, but it does highlight one of the problems with trying to ferret out what malaria is doing in a community. I'll show you here some of the effects of malaria. Causes delayed growth, impaired cognition, and anemia. Iron deficiency causes delayed growth, impaired cognition, anemia. But iron deficiency is it's protecting women from malaria. So if you go in and you're just seeing, do women or children who have malaria, are they different from your control group? If your control group is all iron deficient and they're having the same problems that the people with malaria are having, you're not going to see much impact of malaria. This is true both in children, and it's also true during pregnancy, where a lot of the sequelae of these two. So here's a case where malaria risk can be masked by the effects of another disorder where there's an interaction between these two disorders, and both of them are having adverse consequences for the community. The other thing I want to briefly talk about is the relationship between malaria and preeclampsia. Preeclampsia is a very dangerous condition for pregnant women, characterized by hypertension and protein in the urine. There's an old literature, a very astute obstetrician in Sri Lanka who was there at the time of a dreadful epidemic in Sri Lanka. And what he observed was that during the late stages of the malaria epidemic, there was actually an epidemic of preeclampsia in pregnant women. And he also observed that women that got malaria treatment, the preeclampsia went away. It as well has been observed that preeclampsia goes up and down with rainy seasons, which is the same time that malaria goes up and down. However, when people did detailed studies of malaria's relationship to preeclampsia, they found no relationship when they were looking at parasitemia and comparing that to the risk of hypertension. And actually, when we looked at this in our Tanzanian cohort, we also found the blue bars are the infected women, the red bars are the uninfected women. We found that the systolic blood pressure and the diastolic blood pressure were absolutely the same in these two groups of women. However, this is masked by the fact that malaria has two opposing effects in two different groups of women. What you see here is the percentage of infected women by the red line who have hypertension at different ages on the x-axis. And the blue line is the percentage of uninfected women who have hypertension. So let's look at the blue line first. Uninfected women, it looks like their blood pressure risk is increasing as they get older, which is typical of essential hypertension. But what you can see with the red line is that if you're looking at young, and these are first-time mothers, there is actually a significant increase in the risk of hypertension in women who are infected. The hospital where we worked was not collecting urine proteins. It's not something that's commonly done in many of the hospitals there. So we didn't have that information. We know it's hypertension, but there has been this preeclampsia biomarker identified over recently, sFlt1, and what we found is that first-time mothers who had malaria or hypertension had elevated levels of this preeclampsia biomarker. Those are these bars here compared to the uninfected, non-hypertensive first-time mothers. But when multigravid women had malaria or they had hypertension, the sFlt biomarker is not going up. So this suggests that actually what first-time mothers are experiencing during malaria is probably preeclampsia, although we are now doing prospective studies with urine proteins to confirm that. And what Otis Muhlenbach, the MD PhD student who did this work, found is that this protein sFlt1 that's going up in the mother's blood is actually expressed by the fetal cells. And if you give this protein to rodents, they develop hypertension and proteinuria. And if you give human cancer patients a neutralizing antibody that actually has the same effect as this protein, because this protein binds to VEGF and inactivates it, a neutralizing antibody given to cancer patients has the effect of causing hypertension and proteinuria. So we speculate that actually this phenomenon might be mediating malaria-induced preeclampsia. And why would the fetus start expressing sFlt1, which binds to VEGF? Well, it turns out that the mother's inflammatory cells that come into the placenta to attack the parasite are expressing the cognate ligand, VEGF. And we believe that that's part of an inflammatory process because we know VEGF activates the inflammatory process. I didn't show you the data here because there's not enough time, but there's all the evidence. All the evidence suggests that the fetus does not do well in an inflammatory environment and does everything it can to prevent that from occurring. So it appears to us that what's happening is that the mother's inflammatory cells are coming in to get rid of the parasites and expressing VEGF. The fetus wants to inhibit that inflammatory process, but it does so by secreting a molecule that has the other effect of causing proteinuria and hypertension. And so, again, the way that malaria's effects can be masked is that malaria can have different effects in different groups of individuals, and unless you know how to stratify those groups, you may not see an impact. And this is important because if you look at the horrendous maternal mortality in Africa, in the rich world, there's 20 deaths per 100,000 live births. In Africa, there's 860 mothers dying for every 100,000 births. And these are the major causes of maternal mortality, and three of the most important are all related. And as a final point, an area that we are now getting into is to look at how malaria in the mother is modifying the child's risk of malaria. And I'll just show you this without explaining it further. If the mother has malaria at delivery, it is profoundly affecting the long-term health of her child. The data that I'm showing here just shows the offspring of first-time mothers, the offspring of multigravid mothers, where the red dots are the children that are born to infected mothers, and this is the odds of having parasitemia at any time through infancy. And what you can see is that children born to uninfected women are sort of in the middle, but children born to infected first-time mothers very rarely have parasitemia, and the children born to infected multigravid mothers very frequently have parasitemia. And this looks like it's advantageous, but what we've subsequently learned is that when these children that infrequently have parasitemia do have it, it's a very high density parasitemia. So we're still trying to understand what all the health consequences of this relationship between the mother and her offspring are, but the evidence that we've collected so far suggests that there's an important relationship between the mother's health and the child's outcome. And again, this may help to explain some of the diversity in the experience that children have in malarious areas. So in summary, I told you that malaria vaccines can have different goals because the parasite's life cycle is complex, and by targeting different stages in the life cycle, you can achieve different goals. You can either block transmission to the mosquito, block infection in the liver, or block disease in the blood stage, and I told you a little bit about a vaccine that would target specifically placental parasites to improve pregnancy outcomes. The transmission intensity of malaria affects the disease patterns, and finally the malaria burden is mass. We see that in pregnant women, and we can appreciate it in children's mortality when we eradicate malaria, but we don't have a good understanding for what's going on in kids. And finally, to recognize the people that did the work. I've worked for many years with Mikhail Fried since we both were working at NIH and then Kenya together. Susan Francis and Andrew Olejnikoff worked on identifying what the proteins are that pregnancy parasites express. Ottis Muhlenbach did the work on malaria and preeclampsia in pregnant women. Melissa Bola did the statistical analysis on that data I showed in the last slide about how malaria in pregnant women is related to malaria in their children. TK Mutabingwa is our partner in Tanzania who manages all the clinical research, and Edward Kabyamela is the young Tanzanian scientist who was analyzing the relationship between iron status and pregnancy malaria. Our effort to develop a pregnancy malaria vaccine is funded by the Gates Foundation, and both NIH and the Grand Challenges in Global Health have funded our work to study severe malaria in children to find out what's causing some kids to get very severe disease and die. Thanks for your attention, and we'll happily take any questions. Question?
It sounded like the association with the iron deficiency and illness was an observational study, is that right? And are there any logical confounders to explain that relationship?
The general idea is that the parasite may need iron. So this was the first observation looking at iron status in pregnant women and the risk of malaria. There had been some evidence from women who received iron supplements that their risk of malaria went up. And as I mentioned, there was the study in Tanzania of children. The children that received iron supplements had a greater risk of dying of malaria. So we don't know why that is. There is some suspicion that it's because of the parasite's requirement for iron. Just as naturally, and the anemia of chronic disease is an iron sequestration phenomenon. Thought to occur during inflammatory infectious conditions, and so it's believed that may be part of it, but we don't really know.
About different environments, we know who have access to iron and where that parasitemia is worse.
Right. So the trials with the supplementation were randomized trials, so that they were randomized. Yes.
Hi there. Have you or others performed any type of molecular profiling on the bug in its different environments, or whether in terms of pregnancy malaria versus infant malaria? I mean, can you comment at all? Are there any differences that you do see if that's been done?
So the question is about molecular profiling of the parasites that are infecting pregnant women or children or different individuals with different conditions.
Right. Or even, I guess, maybe the different cycles, I guess, potentially as well.
Yes, so there has been a lot of molecular profiling that went on. A lot of it has been really to look at the diversity of the parasites because, of course, you can get more than one genotype in there. And a lot of the specific genes do undergo a lot of diversification, presumably under immune pressure in the parasites. We ourselves have been looking at transcriptional profiles using microarrays and also protein expression profiles using proteomics tools. And it was that effort that really identified these proteins that are distinctly expressed by the pregnancy parasites, the parasites that infect pregnant women. We are doing this now in children. This is this effort funded by the Grand Challenges program. We're looking to see what the parasites that are infecting children bind to see if the children that have severe, let's say, cerebral malaria, are those parasites binding to something different than the parasites that are infecting children with anemia, for example, and we do have some evidence that supports that idea, but it's unpublished. And we are also now using the microarray tools and proteomics. The problem with kids, studying severe malaria in kids, why it's different from pregnancy malaria, is we could get the parasites from the placenta where it was causing the problem. The parasites we get from the kids come from their peripheral blood. I didn't really mention it, but the parasite has 12 hours of development when it's circulating in the blood before it exports those proteins to the surface that allow it to sequester. So actually the form that you really want to study is sequestered and not accessible to you. The form you can study is the young form, but whereas some of the parasites might be coming from the brain of a child with cerebral malaria, he also has parasites in other organs. So it's a more complex challenge than whereas with the placenta we can get those sequestered parasites and study them very specifically. We don't know yet if there's a molecular marker at the genomic level of the parasites that infect pregnant women. There's been some scant evidence of that, but in essence we can select parasites in the laboratory to bind to that receptor by panning them, and that's actually the model that we work with, is that parasites stochastically express these different PFE and P1 variants. And if a placenta is present, it'll allow the parasite to bind and then multiply. But if a placenta is not present, those parasites will be cleared. It's probably our model is that it has to do with selection on a receptor, the availability and accessibility of, but also whether a host has an immune response to respond to it. We don't know yet if there's something about some parasites that makes them even better at binding in the placenta. We don't have a marker for that yet.